I was talking about our education program. This year we had 19 schools where education in science, technology, engineering, and math was being taught to students. The teachers are being trained so they can teach the students in a proper way. And also we provide science kits to the students so they can do projects because we would like to have uh, project-based learning in Armenia. And uh, and then we have, of course, in the Arakelyan National Lab, we have the detection room where we help the students and the scientists in there to do um, scientific research. And also we have a clean room that we established a couple of years ago, and it is now operational. And we, of course, would like any institute, any university to be able to use that facility. And we also have, of course, ongoing every month this panel discussions and sometimes presentations, depending on the different subjects that we try to present to the public. And today, the uh, panel discussion will be moderated by Dr. Vahan Martirosian. Vahan is a member of our ARPA board, and he works he works at UCLA at the Center for Innovation and Research in Medical Fields as a postdoctoral fellow at UCLA. He has a BS in biology from UC Irvine, and he has a PhD in molecular microbiology and immunology from the University of Southern California. His current interests include development of high throughput screens for personalized medicine and the contribution of metabolism to uh, pluripotent stem cell, which I don't know what it is, differentiation, and global stomata recurrence. So, Vahan, the floor is yours.
Thank you, Dr. Panosian, for that introduction. I would first like to thank everyone for being here, and a special thank you to the ARPA Institute for hosting this important discussion. We're honored to bring together experts and community members to explore the exciting intersection of genomics, history, agriculture, and public health in Armenia. Today's panel will highlight recent advancements in sequencing infrastructure and genomics initiatives in Armenia. We'll discuss how this growing infrastructure is being utilized within Armenia and how it can also be utilized by the Armenian diaspora to advance collaborative research. Key topics include how genomic data is shaping our understanding of Armenian population history, including new evidence challenging the long-standing Balkan migration theory, as well as how bioinformatics is being applied to track microbial antibiotic resistance and improve public health surveillance. We'll also explore efforts to preserve Armenia's agricultural and cultural heritage, such as the Armenian National Grapevine Collection and the role of genomics in protecting indigenous grape varieties, a topic beautifully captured in the recent Grapes of Armenia publication. Together, these discussions reflect Armenia's commitment to integrating cutting-edge science with cultural preservation and public health. We look forward to an engaging and thoughtful dialogue. I would like to first introduce our speakers today. So first, we have Dr. Arsen Arakelyan. He is the director of the Institute of Molecular Biology at the Armenian National Academy of Sciences, head of the bioinformatics group, and co-manager of the sequencing core facility at IMB. With over 23 years of experience in both wet lab molecular biology and genetics and dry lab bioinformatics, his current research is focused on high throughput data analysis, systems biology, and telomere bioinformatics. He coordinates several large-scale genomics projects in Armenia, including the Armenian Genome Project and the Armenian Cancer Genome Atlas initiatives. Next, I'd like to introduce Dr. Levon Yepiskoposyan. He is a senior researcher at IMB and the head of the Institute of Man. He received his Doctor of Science from the Institute of Common Genetics, Russian Academy of Science. He has served as professor at Yerevan State University and IMB. Dr. Yepiskoposyan is also the president of Armenian Anthropological Society and is the head of the Laboratory of Evolutionary Genomics at IMB. He is the author of several books and numerous articles. His main research is in the comprehensive analysis of whole genome data from Armenians, focusing on their genetic history and variation. The main results are that Armenians share significant genetic similarities and that there are genetic input from a Levantine source in the region following the early Bronze Age. Next, I'd like to introduce Dr. Kristina Markaryan. She is the head of the research group on plant genomics at the IMB, a senior researcher at the Laboratory of General and Molecular Genetics at Yerevan State University, and the manager of the Armenian Vitis database, a national platform dedicated to the documentation, characterization, and preservation of Armenia's grapevine genetic resources. Dr. Markaryan's research focuses on the comprehensive characterization of Vitis genetic diversity with special emphasis on molecular fingerprinting, resistance, and molecular breeding, and grapevine adaptation to climate change. She leads several national and international research projects as PI, and her work contributes significantly to the sustainable use of conservation of native grapevine varieties, supporting both scientific progress and the preservation of Armenia's rich viticultural heritage. So I'd like to thank all the panelists for their time here today. Before we have prepared a general outline of the discussion, but if there is any topic that is missed or not covered and everyone else wants to contribute something, I would invite the audience to add their question to the chat and we will get to them at the end. Now I'd like to give each panelist some time to give a brief statement about their work. So Dr. Arakelyan, can you please proceed?
Yeah, thanks very much, Vahan, for this wonderful introduction. I have prepared a couple of slides, just I want to share it with you, just to make my talk more visual. If allow me, okay, I can share my screen.
Yeah.
Are slides visible? Okay, so thanks again to ARPA Institute, Dr. Panelists for organizing this meeting, and thanks Vahan for wonderful introduction of us and our institute and what we are doing there. So I would like to very briefly go through the sequencing infrastructure and genomic projects we are implementing at the Institute of Molecular Biology. So as you may know or may not know, but genomics currently is inseparable in majority of biotechnology, personalized medicine research and clinical implementations, and there are virtually no any field in biological clinical research where genomics hasn't left its footprint. So in our institute, we are involved in genetics and then transforming to genomics for more than two decades, starting from the very early, at the very beginning, from the very generous gift by ARPA of the sequencer machine, which was, of course, the old Sanger sequencing technology, but it actually paved the way of developing and actually giving us the willingness to develop this trajectory of genomics within our institute. And of course, in current realms, genomics has a massive potential of becoming an instrument that will really contribute to personalized medicine, contribute to well-being and healthy aging. So currently we have plenty of projects related to genomics running at our institute. I just briefly mentioned let's say the largest ones. So about mapping of Armenian gene pool and grapevine genome project will be more elaborated by my colleagues Levon and Kristina. and I will briefly run through the rest of those in this presentation. So first and the most maybe important thing now is that we finally were able to create or establish a next generation sequencing infrastructure in Armenia, which is established to promote the advancement of research in areas such as genomics, personalized medicine, environmental science, and what is most important is to foster collaboration between researchers across multiple institutions within Armenia. This project was funded by several, in the frame of several projects, particularly the infrastructure strengthening grant by Research Science Committee of, it's now called High Education and Science Committee of Ministry of Health of Armenia, and sequencing core facilities operated under supervision of our very dearest friend and colleague Jonathan Shook from Pennsylvania University. He is a former technical director of sequencing core facility at UPenn, and Ani Stepanyan, our colleague from Laboratory of Human Genomics. And we hope that this, actually this is now transforming to become a separate unit under their supervision. And we were able to collect all latest technologies in sequencing within this infrastructure, within our core facility. As a main workhorses, we have Illumina NextSeq 2000 next generation sequencer for short reads. This is the lower end of industrial scale sequencing machines. And for long read sequencing, we use now Oxford Nanopore MinION small machines, which allows us to sequence a human genome at coverage of from 6 to 10, with the sequencing depths from 6 to 10. And we also have all other necessary equipment for isolation of nucleic acids, as well as preparing the sequencing libraries, checking their qualities, concentrations, and everything else which is needed in next generation sequencing workflows. With this infrastructure, we are currently performing projects in we are doing whole exome sequencing analysis, we are doing transcriptome sequencing analysis, and are switching to introduction of methylations analysis, metagenomics, and etc., and others. We also recognize the importance of having in place a facility that allows to that will allow to do a clinical diagnostics. So we also licensed our sequencing core facility, part of it, as a clinical laboratory unit that is allowed to do clinical diagnostics works in genomics. So we got a license from Ministry of Health and already performed 20 clinical analysis of whole exome sequencing in Armenia. This infrastructure is coupled with a very large biobank with capacity of more than 40,000 samples. We, in collaboration with Santa Armenia, we creating different biobanks or sample banks for future clinical trials for the range of targeted diseases, as well as for data analysis. We use our compute server, which is equipped with very large memory, operating memory, as well as storage to store genetic data in private way. We use different bioinformatics packages in analysis. The majority of analysis is pipelines are developed by us or built from different components by our bioinformatics team. And for clinical grade analysis, we use a software called Franklin, which is part of QIAGEN now, and it's HIPAA compliant. So basically, it cares very much about protecting genomic privacy. And for population-wide, more research side of the projects, we use Open Cravat toolkit that will be a base for a base interface to present results for a large population scale distribution or analysis of distribution of genomic variants in Armenia. So as a research projects, currently we are one of the priority projects are character or study the genomic variant diversity in Armenian population. We are more focused on clinical side of the work. We are interested in genomic variants that are associated with disease development or modifying the risk for disease in Armenian population, while Levon's team is mostly concerned with the genetic history and evolutionary history of Armenian population. Starting from January 2025, we already sequenced almost 200 genomes with whole exome sequencing technology. And we actually found very interesting patterns of diversity in disease-associated genomic variants across this sequenced sample. For example, we have very high frequency of disease of blood clotting associated disease genomic variants, which is not typical on a global scale. So we aim at sequencing at least 500 samples and then use whole genome sequencing data as a source to refine this data together and create the entire, let's say, Armenian variome or Armenian catalog of genetic variants. The other project is aimed at introduction of long-read RNA sequencing for cancer research in Armenia. We started this project in 2021 and it will finish next year. So the aim is first to bring to Armenia the affordable sequencing technology offered by Oxford Nanopore and also develop bioinformatics pipelines that allow us, coupled with machine learning and AI, that will help to address molecular mechanism of cancer and extract biomarkers for cancer classification, diagnostics, and prognostics. The other also important project with especially for Armenia is understanding the molecular mechanisms that are associated with exposure to environmental metals. Armenia has several mining regions, and we're currently trying to understand, to assess how environmental exposure to heavy metals may affect the health status of the people living there, how it can affect the global gene expression, DNA methylation, and also we are trying to associate these changes with plasma levels of several chemical elements. The other project which shows that Armenia can go way beyond doing genomics projects is studying the effect of space exploration missions on health and performance. So last year in Armenia, there was this Almaty 2024 Mars analog mission, and we were able to collect blood samples from the analog astronauts that were on the mission during 3 weeks. And now we're analyzing their changes in gene expression longitudinally from the very beginning of the mission to the very end of the mission, with the hope to uncover how the immune system reacts to the condition of isolation and heavy extravehicular activities. We're also doing development of new diagnostics approaches for familiar Mediterranean fever using nanopore sequencing technology. We have obtained a patent on this and also have a paper. We introduced a new method for nanopore sequencing that is accurate enough to detect all known mutations in gene-wise in the gene that is associated with development of familiar Mediterranean fever. Last but not least, the project which we are very interested in is related to molecular epidemiology of foodborne infections. This last year's study was involving whole genome sequencing of 44 Salmonella isolates and 17 Klebsiella isolates, shows that there are already several strains in Armenia that circulate, and they are resistant to all antibiotics that are available in Armenia. That's very troubling information, which was, of course, forwarded to the Ministry of Health. And this research is continued in two ways. One is to characterize the overall or map the overall spread of antibiotic resistance mechanisms in different regions, in bacteria collected from different regions of Armenia, as well as to understand what could be the possible non-antibiotic ways of struggling against multidrug-resistant bacteria. For example, whether bacteriophages can be applicable in these scenarios. And this is maybe the first project that we started and successfully completed. It was from 2020 to 2022. We used the nanopore sequencing to understand the distribution of lineages of SARS-CoV in Armenia. And also we were interested to trace the ways how these different variants were transported or immigrated to Armenia. And also we studied the way how host variations in host genome may increase or decrease the risk of infection and severity of COVID disease. So as a future steps, we are willing, and we are now working on this, is to adopt new sequencing and omics technologies to extend sequencing applications, to have more different ways, more different protocols, engage more research groups to work with us. We are now preparing several projects to strengthen the computational data storage resources because we need them more. As a midterm goal, we are very willing, and now we're somehow involved in the process to join with Genomic Alliance for Genomic Health, the section of Human Pangenome project. And we are going to complete our population-wide stage of sequencing and start disease-specific projects. And as another project, we are now doing pilot research on analysis of genomic variants that may define the fitness and predisposition to performance changes or can be used as a markers for modifying the performance in sportsmen. So with that, I would like to thank to all people involved in this in our projects, and also our main funders, which are the State Committee of Science, FAST, and also our industrial partners from David's laboratories. Thank you very much. I hope I didn't over-consume my time.
Thank you, Dr. Arakelyan. That was a great introduction to the Institute of Molecular Biology and all the great projects that you guys are doing. Dr. Yepiskoposyan, would you like to give an introduction? You're muted right now.
Okay. What is this? First of all, I want to thank Jacob for initiating and organizing this very special discussion, and also thank everybody who joined us. Our ongoing project has a title, The Genetic Atlas of Historical Armenian. Actually, we are working on the reconstruction of genetic history of Armenian population, the global population and distinct geographical subpopulations. So this map aims to create a comprehensive genetic picture of the Armenian gene pool, both past and present. So to draw the genetic atlas of present populations, we need to collect DNA samples from Armenians whose all ancestors from maternal and paternal sides derived from distinct region of historical Armenia. It's a very hard work, and we started this work about 10 years ago, more than 10 years ago, and so far we could collect about 3,000, a bit more, 3,000 samples, since it's very difficult to identify our ethnic Armenians whose all ancestors derive from the same region. The second is the genetic atlas of ancient populations, of ancient structures, of ancient strata of Armenian populations. And to do it, we need to collect data from burials. So, and it needs the close cooperation with archaeologists. And fortunately, we could establish very friendly cooperations with our colleagues from different institutions and museums. And now we're proud that we have 3 very valuable papers about the genetic picture of our ancestors, starting the Neolithic times. Based on this unique material, we want to reconstruct in details the complex makeup of the Armenian gene pool and to shed light on our rich and long genetic history, diverse ancestry, and the distinct features that have shaped our genetic identity over millennia. In general, we are trying to address questions that classical Armenian studies cannot. And as an example, neither history, archaeology, or linguistics cannot answer the following questions. When Armenian population started to develop, and how deep are the genetic roots of the Armenian populations? What are the consequences of our contacts with neighbors and with invaders? So, and other questions, etc. So, and we are trying to address these questions, and but we are trying also, we are trying to keep very close cooperations, very close cooperations, yes, and discussions with our colleagues. Since I am not historians, and my colleagues in my lab, they are also have not historical or linguistic background, so we need very professional help from colleagues from different institutions and universities of Armenia to understand and to interpret genetic data in terms of Armenian history. Our project is based entirely on whole genome data, and it means that we need to generate big data, and we need to get this information using sequencing machines. And we started it 10 years ago. And we started it 8 years ago. And that time we didn't have these facilities in our institute. Anyways, the material we are using is a rather big one. I'm telling about the quantity of material. And so we started our collaboration with the Martirosian company, and we generate whole genome data in company. It's much cheaper than if do the same work in Armenia or in Europe. So this collaboration is our collaboration is not only in Europe. Our collaboration is multilateral with our overseas colleagues, and we are currently have very close collaboration with the University of Cambridge in the UK and the Trinity College at the University of Dublin. My former PhD students are working there now, and I have opportunity with my colleagues in Yerevan to expand and to strengthen our ongoing work and the results we are achieving, and to publish it in leading journals. Now, some words about our latest paper published in the American Journal of Human Genetics in the first issue this year, in January issue. The Armenians are characterized by little within population structure, and the special attention was given to Sassoon population, and we show that they derived, they have very specific genetic history, and without any connection with Assyrians, at it was mentioned in some historian papers. Balkan theory on the origin of Armenians has been questioned and firmly rejected, and we hope that this result will be adopted by our Minister of Education, and it will be put in the textbooks in schools and in the universities for students on history, on genetics, and linguistics. I don't know when it will happen, but we still hope. And we also could find the genetic continuity in the region that is constant across time. In the same time, we could identify a large-scale population movement across the Middle East at some point after the early Bronze Age. And this movement brought a Neolithic-like ancestry to the populations inhabiting the regions. Now I'm talking about the event that happened 5,000 or 4,000 years ago. After this event, after this invasion, we had virtually no contact with other populations, and Armenian gene pool was was very stable during last 4 millennia. As a result of this movement, modern Armenians have become genetically altered from their original ancestors. Another key aspect of our project is to engage actively with Armenian diaspora around the world. And using this opportunity, I invite our compatriots to participate in our project, in this initiative, by contributing genetic samples. It's saliva or blood specimens. Usually, in majority of cases, we collect saliva specimens. And we also are expecting that our compatriots will share historical information about their families and the origins of their ancestral origins. Your involvement will greatly support our research efforts. Our next steps, some words about our next steps. We want to reconstruct not only the genetic history of general Armenian populations or distinct groups, but also we want to reconstruct the genetic history of diasporan populations. So maybe next time when we meet, I will have possibility to share the results about the genetic history of Iranian Armenians. It's a very specific and very enigmatic population because there are almost nothing about the source population that contributed to the population of Norjuga Armenians. Summing up, I want to say that Genetic Atlas and our ongoing project is more just a scientific project. It's a journey into our past, a preservation of our present, and a legacy for our future. By understanding our genetic heritage, we will have a deeper connection with our roots and strengthen the bond that unites Armenians across the globe. Thank you very much. I am ready to answer your questions.
Thank you, Dr. Yepiskoposyan. One more introduction. Dr. Markanyan, can you please provide some information about the Vitis Genomics project, please?
I think Dr. Markaryan,
Okay.
she's somehow having problems with the internet, and Arsen is looking into that. So
Okay.
let's start. Arsen, did you find out what happened to Christine?
I'm trying.
Okay, maybe
That's
she'll
okay.
join
Yeah.
later.
Yeah, we can move forward.
Yeah, why don't you start
Yeah.
questioning, and then we can maybe he will.
So, Dr. Arakelyan, one question for you. As you mentioned, genomics has been more and more integrated into medicine and patient care. Are there any established collaborations or pipelines with hospitals in which subsequent sequencing can inform clinical decisions?
Yeah, we actually started to move towards this direction. In fact, sequencing is used as a tool for informed decision on treatment selection or assessment of mutation profile is used like for several years in Armenia, but in almost 100% of cases, the samples were was transported outside Armenia to some clinical labs, and then the report came back. So the idea of what we had is to establish facility within Armenia to shorten turnaround times, first of all, and also to basically get this data in place. And with this in mind, so yes, we started to collaborate with several clinics, especially with National Oncology Center, and also we are collaborating with several physicians from neurology field at the moment, mostly for analysis of germline mutations that are predisposing to the cancer risk, or they affecting or they are related to genetic disorders. But we are constantly looking for growing our current pool of technologies and options for sequencing, and we will soon switch to somatic mutation analysis for cancer treatment selection, and also maybe towards transcriptomic for molecular classification related things.
Fantastic. We'll take a question from the chat. So I think you answered this question, but just for everyone here, is the adoption of liquid biopsies for cancer diagnostics becoming more common in Armenia? And if so, can the Institute expand its sequencing capabilities to help analyze cell-free DNA from these biopsies?
Yes, this is in our plans, and we are currently testing several liquid biopsy protocols for cell-free DNA analysis. As I wrote in chat, we already have good results for testing telomeric sequences in cell-free DNA. And we are now looking for different providers, different manufacturers for nucleic isolation kits, for sequencing library prep kits. And we're also interested, this is a collaboration with Yerevan State University. We are specifically will focus on cell-free mitochondrial DNA in long COVID patients.
It's interesting you bring that up. Recently, there's been a huge boom in mitochondrial DNA research in terms of either its effect on the cell or the transfer of mitochondrial DNA to and from either human, like normal cells to cancerous cells, and how that can affect several other tumorigenic processes. Okay, we can ask some questions for both of you. Why do you think it's important to develop genomics infrastructure within Armenia? I know you touched upon these in some of your introductory speeches, but if you can provide some more information on this, that would be great.
Yeah, that's actually very important question to discuss. So on one side, we know that there are like global providers and global companies that do sequencing, and they do it relatively cheap. And why not to outsource? But on the other side, we faced the COVID as like COVID related shutdown of all logistic routes. And this means that at least you should have something inside your country. You should have the infrastructure inside your country. You should have your stockpiles of reagents inside the country to be able to survive for some times, because this chaos, while this chaos is somehow will get to back to normal. And this was one of ideas what we had in mind by when we will build this infrastructure. The second important thing is having this infrastructure is the only way to train new generation of specialists. So you can outsource as much as you can, but this will mean that you will not be able to prepare people who can deal with this data. And when they need, they will be there for you. And the third one, especially this is related to research projects, we do not benefit as much from sending samples abroad for analysis as from being able to analyze data and enter in collaborations with these added values on top. Because I simply don't want us to turn into a place where we just ship samples outside and some more advanced research groups use them in their research. Of course, collaborations are good, and collaboration should be mutually in mutual way. And having this infrastructure here also already allowed us to engage new partners that have need for sequencing, and they have need for bioinformatics data analysis, which we are doing now within Armenia for the research groups that are abroad. This is maybe the reasons that we want to have it here, like local in place.
Fantastic. Dr. Yepiskoposyan, do you have anything to add? You're muted again. Sorry. Dr. Yepiskoposyan, you're muted.
Sorry.
Yeah.
Usually we use facilities of our colleagues from South Korea because it's much cheaper. I already told about it. But I'm hoping that in some future we will have opportunity to do the same work in Armenia, and it will be very important for us because I don't think that we will have the same problems with as a financial issue. So I'm just hoping.
Is there a benefit to potentially doing whole exome sequencing versus the whole genome sequence?
Yeah, there is a small difference between the using whole genome and exome. Exome usually it's very important and very informative for medical genetics, but we need whole genome sequencing because in evolutionary study we use the neutral positions, not in genes, and that's why the whole genome is especially very important for evolutionary terms.
Great. We'll ask some questions from the chat again. One of the questions I also had is how can we as a diaspora provide samples to you for this project? Is there an online website? Where can we go to find this information?
Uh, you understand that this procedure of sampling is very difficult because very difficult to cover the whole stretch of Armenian highland and from Cilicia to Artsakh and it's very easy or relatively easy to collect samples from Armenians whose all ancestors are from Artsakh, from Ararat Valley, and from Eastern Armenia. And very difficult there is to collect data from Armenians with roots from Western Armenia. And therefore, I need collaboration and need assistance and very strong help from my colleagues, from my friends, from my relatives living in Russia, in America, and in European countries. The only way is to transfer samples is to collect saliva samples. It's a very the easiest way to collect and easiest way to send the samples to Armenia because we need to extract DNA in our facility using our facilities in our lab in Armenia, and then we send the small samples to South Korea to generate whole genome data. And the easiest way is to contact me, and I will explain in details how to proceed and how to uh, order these saliva kits to collect it and how to send it to Armenia.
The other option is when you are in Armenia, and I know that a lot of people traveling at some
Yeah,
point, at
thanks,
some
Arsen.
time to Armenia, to contact us, and we will organize sampling on place. Actually, you will find contact details for Levon and us and me, and also Institute's official emails at our website. And if you just drop us an email saying that we are coming from this time to this, or we basically are interested in providing the samples, we will organize sample collection and transportation to the Institute.
Great, thank you. Dr.
Thank you.
Yepiskoposyan, you mentioned about how it was difficult to trace some of our roots in Western Armenia, and we obviously know that's where a lot of our ancestors perished from the Armenian genocide. Do
Yeah.
you think it's possible to estimate the impact of the genocide on the diversity of the modern Armenian gene pool?
Oh, it's very interesting. Not only interesting, this maybe this one of the principal questions I'm trying to address, and but we need a very special collection of fossil data. We need collection. We need the information about the original diversity, genetic diversity in population before the genocide. And now I'm thinking about it, and there are some possibilities. I don't share this information openly, but definitely we have some possibilities to collect this kind of data and to extract the squeeze necessary information to address this question.
Fantastic. Thank you. We'll take some more questions from the chat. Dr. Yepiskoposyan, what do you expect from the Iranian Armenian project? Mixed genes with different ethnic groups?
Of course not. There are some information, historical records about the event that took place uh, four centuries ago, in the beginning of 17th centuries, when Armenians were forcibly transplanted to Iran, to the central region of Iran. And but the authors of these records historians, they don't mention what regions they are from exactly. And relying on our data, mainly based on Y chromosome data, in other words, the patrilineal genetic history, we could conclude that there are not roots or evident roots of or traces or signals of Armenians from eastern part of Armenian highland, from Sunik and from Artsakh. So we want to refine this result or this conclusion and to get more information, overwhelming information, deep information about this preliminary conclusion. So we need to collect more data from Iranian Armenians, especially descendants of Armenians forcibly taken by Shah Abbas I, and to address this question based on not historical data, but on genetic data.
Great, thank you. So obviously, sequencing and genomics in general creates massive amounts of data that no individual on their own can parse through or integrate. Is there any potential for AI involvement in analyzing this data, or is there any work being done in that regards? Dr. Arakelyan, we'll start with you.
Yeah, actually, AI is let's say started to become a tool for analysis of genomic data. We already know that Google released latest version of its AI-based genomic variant detection pipeline, which is now is extensively tested against the gold standard pipeline, which is developed by Broad Institute. And it seems the results are very promising. So AI will be helpful in the fields where it's really gigantic amount of data already available. For example, analysis of or identification of mutations within sequences. It can be done very well because from each sample you get about 4 million, 5 million points of data, and you have already around several 100,000 of people data available. So, but when we are talking about, for example, disease classification or treatment selection, at the moment, AI did not, these methods did not perform well, relatively well, because of severe limitation of samples available for training and testing. And what is even more affecting is severe heterogeneity within the data. So two different labs will collect same patients or same data differently, and sometimes it's really impossible to merge this data together to get one bigger pool. And this is what seriously limits advance in biomedical research. If you just see, we have very well results in image processing, we have very well results in natural language models, in structural bioinformatics drug design, but when it comes to genomic data, still there are not much instruments available for that. And this is because these inherited problems in data collection and data synchronization, but I'm sure that within recent years, this problem will be solved and we will have much deeper integration of AI within genomic tools.
Great. Dr. Yepiskoposyan, do you have anything to add? You're muted still.
Fault. I have no idea about it because I'm trying to use more effectively the current possibilities and the current possibilities of genomic theory and practices, and we have very, very complicated instruments, tools, mathematically based tools to analyze the whole genome sequencing data. So if some possibilities or new possibilities of AI will be helpful on this matter, I will be only very, very glad.
Great. This question for you from the chat. Is there evidence of Armenian genome migration from Africa to Armenia?
It's not, it's not the, what is a scientifically based question, because all ancestors of modern populations out of Africa or beyond Africa, they derived from Africa. So I can say that exactly our ancestors, deep, deep, deep ancestors, about 40 or 70, no, 60, 50,000 years ago, they came from Africa, and we can trace back this connection, and but it doesn't relate to the Armenian genetic history, to the genetic history of Armenian population. So it no sense to reconstruct this connection.
Great. An overall question for both of you. Are there any current challenges related to the cost or availability of essential supplies, such as sequencing kits or reagents, or even storage for data? I know you're generating tons and tons of data, so is there any issue with the storage in general?
Yeah, actually, that's from the beginning. So, of course, due to logistic specificities and geographical location of Armenia, we have a bit we're in a bit difficult situation for in getting reagents and supplies for sequencing compared to the European or Asian countries. For example, when we order a kit for sequencing kits or flow cells, at the first several months, they traveled from the storage in Singapore to all the way to the Frankfurt and then to Yerevan. Because there was no direct flights from Singapore to, or at least connections that coming to Armenia from like Asia. But our partners at Illumina they are really willing to help us to solve these issues, and we now have much better ways of getting reagents. Though of course they cost at least 1.5 times more than in the Europe or in the US. Regarding the data, it's another issue because we really generate a lot of data during every run, and we have problems like several separated problems. We have the need for computational space where this data will be loaded and analyzed, and then where it will be stored, and the analysis data results should be separated from raw data. Raw data also should be backed up somewhere else, just in case. And if we follow the guidelines, it should be then re-back up in another place, just to make sure that we will never lose it. And this requires a huge like storage space. But we applied and got a grant for buying a new storage server, which will bring our overall capacity to some few petabytes, which will be enough for some time. And also we have access to Aznavu supercomputer for where we can use it for like computational heavy analysis parts, like whole genome sequencing analysis, because whole exome we can do like routinely on our servers. So that's the general way of what we are doing now.
Dr. Yepiskoposyan, do you have anything to add?
Well, I totally agree with Arsen, because we have the same challenges the same issues, but just in general, Armenian legislation and these norms and regulations, they are not favorable to our academic community. Very difficult to, and it's very costly to purchase some instruments, and especially very costly to order this work and whole genome sequencing in South Korea, because our legislation, they ask us additional taxes if you order any work or any services outside Armenia. So we need to think about it, and we need to fix it. Otherwise, we are trying to find tricky ways to bypass these regulations.
Great. As many of us are obviously focused on is the student engagement, and you, Dr. Arakelyan, you brought this up in trying to keep the sequencing in Armenia is to pass on that knowledge to the next generation. How are the students being trained or engaged in these projects and techniques, and are they given their own research projects for their own hands-on experience? How is it going in both of your labs?
Yeah, basically, engaging young generation of specialists in research is one of our top priorities. We have several like initiatives aimed at bringing young scientists to work with us. We started to work with them when they are students. We have our joint educational departments with International Scientific Education Center of Academy of Sciences, and also a joint department at Russian Armenian University. Our researchers teach courses at American University, teach courses at Yerevan Medical State University, teach courses at Yerevan State University. So basically, we are trying to communicate with students at their very early years of their programs. And this actually allows us to bring a lot of young people at the institute. The average age at the institute is about 40. It means that there are we have a lot of masters and bachelor students doing their theses, working that join to different research groups as research assistants or or junior scientists. And the idea is that we are not afraid to give them hands-on work. So we know that if you want to someone learn something, they need to train and they need to spoil something. It's not possible to do a good job without deep understanding the process and also breaking several pipettes during the work. We are okay with that. And what we do now, we have several seniors that are very well specialized in different sequencing protocols. And during each sequencing run, first, our young specialists basically they familiarize themselves with the protocols, and then they do step by step under supervision of the senior person. And they do it as long and as much as is needed to them to become a professional or do it in a professional.
Dr. Yepiskoposyan, do you have anything to add?
Yeah, we don't have sequencing facilities in our lab, so the only work we can teach is the statistical genetics and some modern tool that we use to analyze this whole genome sequencing data. So it's not so difficult because we have close contacts with my former PhD students, and the possibilities of web possibilities also are very important and very helpful to spread this knowledge and to disseminate these these skills among young students.
Fantastic. So another question for you from the chat. Do you extract ancient DNA from archaeological sites? And if so, is this extraction work being done in Armenia, or do you send the materials to the US or Europe for preparation and analysis?
It's a good question, and I wanted to stress this point in my introductory word, because it's we need to be very careful while sending our ancient DNA samples to beyond Armenia, because I'm confident that we need to analyze ourselves, and we need to explain and interpret ourselves the results of ancient DNA analysis. And unfortunately, we have some very bad, very sad events, sad results, sad events, yes, when Armenian ancient DNA data are analyzed by not Armenian geneticists, and because these data or these samples were sent by Armenian archaeologists uh, beyond Armenia, out Armenia, and to different countries. And in my case, I always demand, I always ask my colleagues, my collaborators in the United States and especially in Europe, that the ancient DNA data samples should be analyzed by my students, by my young colleagues, and the data should be interpreted by Armenian colleagues, by Armenian geneticists and anthropologists. So this is the very delicate issue I want to stress.
Great, thank you. One question for both of you is, are there any pressing needs in the genomics research community right now? I know we spoke about a lot of them, but something in regards to infrastructure, education, or finance, potentially, something, one or two points that both of you can make. What's the most pressing need right now in this community?
Oh, okay. Levon, would you like to go first?
Oh, I need to figure out what to answer.
Okay, so if we are talking about Armenian genomics community, the pressing need is the first maybe awareness of the options that we have in the country, within the country, and we're trying to disseminate the information as wide as possible, and to like basically offer different research groups to become users for the facility. The other thing is, at the moment, it's important to have updated legislation on management of genomics resources. This is of uttermost importance, because for many years, these samples were basically taken out from Armenia. Especially, I'm talking about plant biodiversity samples, and were used in many like very important and high impact projects without proper acknowledging of their origin, without proper acknowledgement and benefits to Armenia as a country from which these samples were originated. In this regards, I very much value what Levon and Christina are doing, because although they use their wide collaborators networks, and use a lot of or they are doing a lot of research outside, but they very well are able to protect and place their role in the research and in following publications. That's an important thing that basically gives additional credit to Armenia as a place for cutting edge genomic research and cutting edge and the source of unexplored biodiversity.
Dr. Yepiskoposyan.
Yeah, again, I totally agree with Arsen's concerns. But in our case, it's a special area. It's an area which has very close relations with history, with our roots, with Armenian language, with our customs. So it's a very complicated area of Armenian studies, and unfortunately, we don't have possibility to spread and to disseminate scientifically based information to their people, to starting from ordinary school. So this is maybe a bit strange concern, but it's a real concern. And I'm trying to be very active on this matter, trying to disseminate knowledge. But sometimes I need the assistance of journalists to spread it and to give me floor, to give me opportunity to have more audience, general audience, to teach them the basic knowledge on Armenian genetic history.
Great. So we've spoken at length with the infrastructure in Armenia. One of the questions that I have, and it's of personal interest to me as well, is how can this growing infrastructure be leveraged on the international scale, particularly through partnerships with diaspora researchers? Dr. Arakelyan, would you like to take that first?
Yeah, thanks. It's very interesting and important question. Actually, we were building our infrastructure with having this in mind. That again, we have two main routes or pathways in genomics research. One is a wet lab, and one is dry lab or bioinformatics research. So currently, we are able to actually offer a full package of our services to research groups worldwide. And we already have collaborations established on this particular infrastructure. We are now collaborating with our very good colleague, David Lukasyan from Mount Sinai University. We are doing a project related to space cosmic irradiation effects on cardiac toxicity. And he actually sent his irradiated mouse samples to Armenia. So we are planning to do methylation sequencing analysis, and also we use data that he obtained before with other company in the US for bioinformatics data analysis. We have our collaborators at Armenian Bioinformatics Institute have a lot of collaborations with international companies and startups, and also companies established by Armenian diaspora researchers. And they offer bioinformatics data analysis. On the other side, we are now collecting a very interesting longitudinal data set of inflammatory bowel disease patients with aim to characterize their cell-free DNA content and link it to the microbial community analysis in gut and analysis of immune response profiles. we believe that these data sets and also samples together can be interesting for researchers that are involved in IBD research in general. So yes, we are looking forward to extending our collaborations, and we actually have various facilities and various competencies to initiate and complete it.
Great, thank you. Dr. Yepiskoposyan?
Yeah, unfortunately, our area is not so dense as molecular biology, and in the area of molecular anthropology, there are only maybe 2 or 3 Armenians working on reconstructing Armenian genetic history. Unfortunately, I couldn't successful trying to establish contact with them, especially with the guy working in France. So my experience is not positive. And in the same time, I'm working very closely with my former students, currently working in different countries, in different leading centers and universities in Western Europe and in the United Kingdom and Canada. United Kingdom and in Canada. So I am very satisfied, and that I'm working not the real Armenian diaspora, but with my former students and PhD students working in different countries, and they have opportunity to contact very closely with Armenian communities in the countries of residence.
Perfect, thank you. We'll take some more questions from the chat. So, Dr. Arakelyan, this one's for you. Are there any avenues in Armenia where the discoveries found through omics are then translated into drug therapy development?
There are avenues, but it never happened at the moment. So we expect that we basically build an opportunity for that. And we hope that there will be startups or research groups that will use this infrastructure for this purpose. We already see several startups that grown very efficiently in particularly from our institute as startups that deal with drug design, but they mostly are in the field of AI-based intelligent drug design from structural bioinformatics point of view. But I think that at some point there will be genomic startups that will utilize our infrastructure for basically more like genomic-based drug and therapy development.
Is the data that you have or collected available to the overall general public or scientific community in Armenia?
It will be open soon, so we are waiting to cross 200 sample threshold to reanalyze this data, prepare manuscript and portal that will be available to entire scientific community with averaged densities or frequencies of variants. Of course, will not be raw data, but we also are developing processes and ways of providing access to raw data based on the commonly accepted standards in the field.
Great, thank you. Dr. Yepiskoposyan, I think these next few questions will be for you. One question is there a pure Armenian genes or genome?
Oh, it's maybe the biggest issue for me, because always I have questions from different peoples, sometimes even from physicians, from doctors, from educated people that there should be a special, specific, distinct Armenian gene that is responsible for our peculiarity, for what is to be genius, and so, etc., etc., etc. Of course, not my answer is that if we have a special distinct gene, so we need to separate. We have to distinguish separate species, not Homo sapiens but Homo sapiens armenicus, and it doesn't exist. So the distinct gene also doesn't exist.
Great.
If I may add, so basically what I say in these situations, there is no Armenian genes, but there can be Armenian genome, in which the distribution of different genetic variants in genes and their frequency will be a bit different from surrounding or neighboring populations. And this what we are actually trying to study now.
This is diversity. It's diversity. So where we always study diversity, but not the peculiarity in terms of separate, distinct species.
Great, thank you. There's a question here from iMac27. Not quite sure. It says Hamshen CS. I'm not sure what the
Yes,
question
this
is.
So if iMac27 is Hamshetsi, please contact Levon Yepiskoposyan, because Hamshen is Levon, if you can.
Hamshensis. Hamshensis, it's a geographic subpopulation of Armenians, and we're trying to reconstruct their genetic history based on whole genome study. So far, we could reconstruct their patrilineal history and found that they have nothing to do with the Turkish tribes, which invaded to Armenian plateau some 1,000 years ago. And so we could prove the Armenian roots of this community, of this subpopulation. But we want also to answer one additional question about the genetic proximity between Christian Armenians and Islamic Hemshales, currently inhabiting the Pontic region of Turkey. So, and we hope that next year I will be able to share this information with you.
Great, thank you. Another question here is, when there's research done in Iran, it is always focused on Armenians who were relocated by Shah Abbas. How about the indigenous Armenians from north of Iran?
Yeah, the work related to Norjuk Armenians was done about 10 years, maybe a bit more years ago, and it was done by my PhD students, originally from Tehran, and relying on this, based on these results, I told you that we couldn't find any traces, any visible traces of Artsakh or Sunni Armenians in gene pool of Isfahan Armenians. But unfortunately, the original population, Armenian population on the northern regions of Iran, they very difficult to collect data there because the villages are abandoned, and I had the opportunity to travel several times to the northern regions of Iran, and we met only maybe a couple of families in these abandoned villages. So very difficult to get enough samples to make evidence-based inferences.
Great, thank you. One question, last one. What are the oldest DNA samples you have extracted from?
It's about 8,000 years. It relates to Neolithic times, and this sample was collected, was dug by my team in Karabakh, in Karintak cave. Unfortunately, we left this site, and we also had other three or four Neolithic samples uh, acquired in different sites in Armenia, in the Republic of Armenia. So we have only 4 or 5 samples related to Neolithic times in Armenia. Unfortunately, it's not enough to get to a whole picture or diversity of this stratum in our history.
Another question, drug development for what used to be called, quote unquote, the Armenian disease.
Yeah, this is another project we are involved in now. We are now creating a patient-specific bank for induced pluripotent stem cells from Armenian disease patients. Actually, the official name of the disease is familial Mediterranean fever disease, and this is genetic disorder associated with the mutations in MEF3 gene. So we are currently creating this biobank for patient-specific stem cells for 2 purposes. One, we want to test different small chemical compounds as a potential substitutes for colchicine. But the other project or aim is more ambitious. We want to develop genetic therapy for this disease. We want to create a CRISPR or genome editing based construct to either correct mutation or perform whole gene replacement. We are now just very early steps of the project. We don't have any like sound results, but the idea is to work in this direction to have like virus, some AAV-based delivery system that will deliver construct within immune cells and perform gene therapy.
Fantastic. I think that's the last question we had in the chat. Does anyone else have any last questions that they want to ask?
Levon wants to say something.
Yeah, I just want, if I may, yeah?
Of course, of course.
Yeah, I want to add some words to the previous questions about the Armenians from northern Iran. I forgot to mention that in our project, a genetic atlas of historical Armenia, we have 2 distinct regions, and from northern Iran, it's Maku, and the second one is Khoy-Salmast. So these regions will be also included, but they are only descendants. We are collecting DNA samples from descendants of people who lived maybe a century or 2 centuries ago in the northern regions of Iran. So in general, the genetic peculiarities or genetic features of these 2 populations will be included in our project.
Great.
Great, fantastic. Thank you so much. With that, I would like to thank the ARPA Institute for hosting the discussion, the panelists for their time and fantastic answers, the audience for their attention and for their great questions. And I'd like to wish everyone a great rest of their day. Thank you so much for everything.
Thank you, Vahan, and thank you, Levon and Arsen, for your excellent presentation. Unfortunately, there were 2 others that were going to join, and actually one of them was with us in the beginning, but somehow she disappeared. Christine Margarian, She was going to talk about a very interesting subject which she's researching on, about new kinds of grapes that are, from Armenia and only in Armenia. So, but unfortunately, she couldn't join us. And also, Lidit Nersisyan was going to talk about her work at Armenian Bioinformatics Institute that she heads. But she was not feeling well, so she couldn't join as well. But thank you, everybody, for joining us and for the excellent presentations. Goodbye.
Thank you.
Thank you. Thank you very much. Thank you, Dr.
Goodbye.
Panelists. Thank you, Vahan, for this opportunity. And thanks for the audience for insightful questions and being with us till the end of the panel.
Thanks.
Thank you